Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. Today we're diving into something pretty fundamental, how our cells actually make proteins. And here's the kicker, the really surprising thing. For ages, we kind of assumed that if you knew how much messenger RNA a cell had, you basically knew how much protein it was making, what the cell says. But this paper we're looking at today, it showed that what a cell says, the mRNA, and what it actually does, the protein it makes, can be wildly different. We're talking about a technique that lets you see protein synthesis with, like subcodon precision, seeing exactly which letter of the genetic code machinery is on. It revealed things like molecular traffic jams and even these secret starting points for making proteins that nobody knew were there.
That's exactly right. And it's such a critical point because it shows how one sort of methodological breakthrough can just completely shift our understanding of a really core process in biology. This isn't just about knowing, which genes are switched on. It's about how they're being read, how the instructions are actually used. And it turns out that process is incredibly dynamic and way more regulated than we thought. So this deep dive is really about how this method, ribosome profiling, just redrew the map of gene expression.
Okay, let's unpack this then. Our mission today is to dig into this game-changing 2009 paper from Science that introduced ribosome profiling. We'll walk you through how they did it the ingenious method and then the, mind-blowing stuff they found, specifically in budding yeast, which is a pretty standard lab organism.
Yeah, and maybe just a little context first, because to really get why this was so big, you need to know what came before.
Good idea.
So before this paper, biologists had decent tools for measuring mRNA, right? Like microarrays, early sequencing. We could get a parts list, genome-wide. But watching protein synthesis itself, seeing where the ribosomes, the protein factories actually were on the mRNA blueprint, how fast they were moving, that was like trying to see individual cars on a highway from like an airplane. You get the general traffic flow, maybe, but no details. Older methods like polysome profiling, they gave a blurry average. Reporter assays, they only let you look at one gene at a time, maybe two. What was really missing was an unbiased way to see ribosome activity across all genes at the level of single nucleotides, the individual letters of the code. That was the key to really understanding how genetic information flows.
Right, so they needed to get down from the airplane view to being right there on the highways, seeing every car. How did they build this molecular microscope then? What was the trick?
Well, the core concept is actually pretty elegant. It hinges on a simple physical fact. When a ribosome is actively translating an mRNA molecule, it physically covers up a small piece of it, about 30 nucleotides or so. And this bit of mRNA that's covered, it's protected from being broken down by enzymes that chew up RNA. So these protected fragments, they call them ribosome footprints.
Like a little shield the ribosome carries around while it works.
Exactly like a shield.
Okay, so how did they isolate just those shielded bits? The workflow sounds really clever. They started by gently breaking open the cells, right? Carefully. So the ribosomes stayed attached to their mRNAs.
Yep, preserving those active translation complexes was crucial.
Then they added an enzyme, an RNase you called it, like molecular scissors.
Precisely. This RNase chews up all the exposed mRNA, everything that isn't physically shielded by a ribosome.
Leaving behind just those tiny 30 nucleotide footprints.
Just the footprints, then they had to carefully fish those out.
And here's the really key step, converting those little RNA footprints into DNA so they could use modern sequencing.
Exactly. They created a DNA library from these RNA footprints, which could then be fed into deep sequencing machines. These machines can read millions and millions of these short sequences incredibly fast.
So you get a massive list of all the footprints that were in the cell.
Right. But getting there wasn't trivial. They had to really optimize the enzyme digestion too much, and you damage the footprints too little, and you get background noise. And importantly, they developed a new way to prepare that DNA library. Older methods often use specific enzymes, RNA ligases, that apparently introduced, and I quote, large distortions in the data. basically skewing the results.
Ah, so artifacts. Things that look real but aren't.
Exactly. Their custom method avoided those problematic steps, which was absolutely key for getting accurate quantitative data. You need to trust that what you're measuring reflects reality.
Okay, so they can accurately isolate and count these footprints. What's the real power there? What does counting them tell you?
Well, two things. First, by seeing which footprints are present, you know which mRNAs are being translated. That gives you a measure of mRNA abundance, similar to older methods, but potentially more accurate. But the real breakthrough is that the number of footprints mapping to a specific region tells you the density of ribosomes there, how busy that part of the mRNA is.
Ah, so it's not just if it's being translated, but how much. Like measuring traffic density on that highway.
Precisely. It's a direct quantitative readout of translational activity, location by location, across the entire genome.
And then they checked this, right? Made sure it was reliable.
Oh, absolutely. They did rigorous validation. They showed extremely high reproducibility between biological replicates, meaning if they did the experiment twice, they got almost identical results. An R-squared of 0.98 for mRNA abundance, which is, nearly perfect correlation.
That's impressive.
And it lined up pretty well with older microarray data, too, but their ribosome profiling data actually correlated better with known protein levels, suggesting it was giving a more accurate picture of what was really going on. Plus, their method handled trip bits of RNA structure better and kept track of which strand of DNA the RNA came from, which was important for precision.
Okay, so they built this incredibly precise tool. Let's get to the payoff. What did they actually see when they pointed this thing at yeast cells? What were the first big aha moments?
Well, one of the most striking, defining features they saw immediately was this really strong 3-nucleotide periodicity in the footprints. Meaning, when they mapped exactly where the footprints started on the mRNAs, they weren't random. They overwhelmingly started every 3 nucleotides. Click, click, click. Along the message.
The codon triplet.
Exactly. It was the unmistakable signature of ribosomes moving along the mRNA and reading the genetic code in its 3-letter words, the codons. It was beautiful, direct, genome-wide proof that the method was capturing the actual reading frame of translation.
Wow. So that highway analogy again is like suddenly seeing not just cars, but seeing them perfectly aligned in their specific lanes, moving one car length, one codon at a time. And they said, 75% of the main footprint size started right on the first letter of the codon.
Yeah, something like that for the 28 nucleotide fragments. That level of precision was just unprecedented. It opened the door to studying really complex things genome-wide, like programmed frame shifting, where ribosomes deliberately slip into a different reading frame.
Okay, so precision confirmed. What else? What about the overall picture?
Well, they measured translation rates for, I think it was nearly 5,000 genes. Basically, most of the yeast genome was really high accuracy. And what jumped out was this huge range in how efficiently different genes were translated. We're talking about 100-fold difference between the most and least efficiently translated mRNAs.
100-fold. Okay, this brings us back to that initial point. This really challenges that old idea, doesn't it?
Yeah.
That mRNA levels alone tell you about protein production.
Absolutely. It demolishes it, really. They found that differences in this translational efficiency, how many proteins you get per mRNA molecule, contribute massively to the final protein levels. Their data showed mRNA abundance alone is, as they put it, an imperfect proxy. When they correlated things, translation rates measured by ribosome profiling predicted actual protein abundance much, much better R-squared of 0.42 versus only 0.17 for mRNA levels.
Wow, that's a huge difference. Almost 3 times better prediction just by looking at translation.
Exactly. It proved that a massive amount of regulation determining protein levels happens after the mRNA is made, at the level of translation itself, stuff we just weren't seeing clearly before.
Okay, but it gets even weirder, right? You mentioned traffic jams. They found ribosomes weren't moving smoothly along the whole message.
Yeah, that was another huge surprise, a really general feature they uncovered. When they averaged the ribosome density across hundreds of genes, they saw something consistent. In the first 30 or 40 codons right near the beginning of the protein coding sequence, the ribosome density was significantly higher, about 3-fold higher than further downstream.
So like a pileup right at the start.
Kind of like a bottleneck, yeah. Ribosomes seem to accumulate there before spreading out and moving more consistently along the rest of the gene. And this wasn't just in a few weird genes, it was a general trend, regardless of how long the gene was or how heavily translated it was overall.
So it's not a smooth on-ramp onto the protein synthesis highway. There's like a slow merge lane at the beginning for everyone.
That's a great way to put it. And discovering this actually solved an old puzzle. People had noticed before that shorter genes seem to have more ribosomes on them per unit length.
Ah, because a bigger fraction of their length was this high-density start region.
Exactly. When they corrected their protein synthesis estimates to account for this initial ribosome surge, the correlation between their translation measurement and actual protein abundance got even better. Went up from 0.42 to 0.60.
Okay, so understanding that initial bottleneck gave an even clearer picture. What causes it, though? Slower movement at the start.
That's what they suggested. It could be that initiation is fast, but the initial elongation steps are slower, or maybe there's more pausing or even ribosomes falling off early on. It points to complex regulation happening right at the beginning of translation.
Okay, so we've got incredible precision, proof that translation efficiency varies wildly, and this weird bottleneck at the start of genes. This tool is really peeling back layers. What other secrets did it uncover about where translation is actually happening?
Right, so beyond just how much translation, they could pinpoint exactly which parts of the RNA molecule had ribosomes on them. And while, okay, almost 99% of the footprints mapped where you'd expect to, the known protein coding regions that still left over 56,000 footprints mapping elsewhere, these weren't just random noise. They often clustered in specific locations.
And where were these unexpected clusters?
A lot of them were found in the 5' untranslated regions, the 5 UTRs.
Okay, so that's the bit of mRNA before the main protein coding sequence starts, usually thought of as just regulatory landing pads.
Exactly. Now, some translation there was already known to happen. There are things called upstream open reading frames, or uORFs. These are short little coding sequences in the 5 UTR that can make tiny peptides and often regulate the translation of the main protein downstream. And their data picked these up beautifully. They identified over 1,000 potential uORFs, confirming lots of known ones, like in a gene called ICY1.
Okay, so uORFs explain some of it, but then came the really wild part, right? Finding translation that didn't start with the normal start signal, the AUG codon.
Yes, this was really counterintuitive. The AUG codon is like the universal start here sign for protein synthesis. But they found clear evidence of ribosomes translating regions in 5 UTRs that had a stop codon, but no AUG start codon anywhere upstream.
How's that possible?
Well, they looked closely at specific examples like genes called PRD2 and PDR5. In both cases, the pattern of ribosome footprints strongly suggested that translation was actually starting at a uUG codon instead of AUG.
UG, so a different three-letter word could act as a start signal.
Apparently so, at least in certain contexts. This was a massive finding. It meant the rules for starting translation were more flexible than we thought. It wasn't always AUG.
So some ribosomes are basically ignoring the main green light, AUG, and just starting at a different signal if the conditions are right, taking an alternate route.
That seems to be the case. They dug deeper and found 143 of these non-AUG-initiated uORFs with solid evidence of translation. And remarkably, these accounted for about 20% of all the ribosome footprints they found in the 5 UTRs. And some of these non-AUG start codons, especially ones just one letter off from AUG, were actually translated pretty efficiently, sometimes even better than some AUGs. It pointed to this pervasive, regulated use of non-canonical start sites.
Mind blow. Okay, so this method isn't just mapping the known roads, it's finding hidden side streets and shortcuts. What about seeing things change over time? Can it capture how cells adapt, like under stress?
Absolutely. That's another huge strength. Because it's quantitative, you can compare samples under different conditions and see exactly how translation changes. They did exactly this, looking at yeast cells under amino acid starvation, basically, when the cells are stressed because they're running out of protein building blocks.
And what happened? Did translation change much?
Oh yeah, big changes. They found about a third of all genes showed significant changes in their translational efficiency during starvation. Nearly 300 genes had their efficiency changed by more than twofold, up or down.
Any patterns?
A major one was that genes involved in making ribosomes themselves, ribosome biogenesis genes, were strongly downregulated translationally. Makes sense, right? If you're starving, you slow down factory production. The statistics on that were overwhelming, like p less than 10 to the minus 40. A very clear, strong signal.
Okay, cells slam the brakes on making new protein factories. What about specific genes involved in the stress response itself?
Yes. And here they saw a classic example beautifully illustrated. There's a famous stress response gene called GCN4. Its protein helps the cell cope with starvation. They saw a massive 7-fold increase in the translational efficiency of GCN4 during starvation. This gene was known to be regulated at the translation level, but older methods like polysome profiling didn't quite capture the full extent of this dramatic induction. Ribosome profiling nailed it.
And GCN4 is regulated by those upstream uORFs, those little sequences in the 5UTR we talked about, isn't it? It all connects.
It connects perfectly. GCN4 has multiple uORFs. Under normal conditions, ribosomes translate the first uORF, then they usually reinitiate at the later uORFs, which prevents them from ever reaching the main GCN4 coding sequence. So, little GCN4 protein is made. But during starvation, a key signaling pathway changes how reinitiation works. Ribosomes still translate uORF1, but then they're much more likely to scan past the inhibitory uORFs 2, 3, and 4, and finally reach and translate the main GCN4 coding region.
So the uORFs act like switches, and starvation flips the switch to ORF for GCN4 production.
Exactly. And the ribosome profiling data showed this shift. Visually, you could see the ribosome density decrease on the inhibitory uORFs and pile up on the main GCN4 start codon during starvation.
That's cool, but wait, didn't they also find something new in GCN4 related to that non-canonical translation?
They did. They found evidence for new, previously unannotated translation initiation, likely non-AUG, within the GCN4 5UTR itself, and this unexpected translation event was also enhanced during starvation.
So under stress, the cell isn't just using the known GCN4 switch, it's maybe using other non-standard ways to boost related processes too.
It seems like it, and this fits a broader pattern they saw. Remember those 5UTR footprints? During starvation, the overall number of footprints coming from 5UPRs increased massively, about 6-fold. And crucially, the footprints mapping to those non-AUG-initiated uORFs showed a particularly dramatic increase in occupancy. Sometimes, translation from these non-AUG sites became incredibly prominent during stress, even exceeding the translation from some normal coding sequences.
Wow, so it's like the cell gets less picky about where it starts translation when it's under duress. The rules relax.
That's the interpretation, yes. It suggests a general relaxation in the stringency of start codon selection during stress. This might be mediated by a factor called EIF2 alpha, which is known to be involved in controlling translation initiation during stress. The cell basically says, okay, times are tough, let's be a bit more flexible about finding the instructions we need.
Okay, this technique is just incredible. It goes way beyond just counting mRNA. So let's zoom out. Why is this 2009 science paper considered such a landmark? How did it really change the game?
Well, its impact was huge on several levels. First, the theoretical impact. It fundamentally reframed how we think about translation. It wasn't just an assumed follow-on from transcription anymore. It was revealed as this incredibly complex, spatially resolved, genome-wide process. It established translational efficiency as something you could actually measure accurately, genome-wide, not just infer. And it showed just how often mRNA levels don't predict protein levels. Plus, it revealed that things we thought were rare or weird, like complex ribosome density patterns along a gene or widespread initiation at non-AUG codons, are actually common fundamental features of translation. That's a paradigm shift.
So it changed the textbook picture. What about the technology itself?
Technologically, ribosome profiling, or ribosec, as it's often called, now became a cornerstone method in molecular biology. It's incredibly versatile. People immediately adapted it to map exactly where translation starts, to measure how fast ribosomes move, to study stress responses in detail, to find hidden small proteins encoded by uORFs or other non-canonical regions. And a huge advantage was its genome-agnostic nature. You don't need special tags or engineered cell lines. You can, in principle, apply it to almost any organism or cell type. That was revolutionary for unbiased discovery.
And the applications, where has it led?
It's unlocked discoveries all over the place. Understanding how uORFs control gene expression, discovering entire new classes of small translated peptides, figuring out how viruses hijack the cell's translation machinery, understanding how certain drugs work by targeting the ribosome. It's used constantly in basic research and even in developing new therapies.
It really does sound like they built a kind of time machine or maybe a high-resolution map, letting us see these fundamental molecular processes with completely new eyes.
That's a good way to think about it. It revealed unexpected complexity everywhere they looked. Even just the variations in ribosome speed along a single gene suggests the ribosome itself might exist in different functional states, doing slightly different things. And it opened up whole new questions about stress responses, about defining the complete set of proteins an organism can make, the proteome, including all those non-canonical ones. It gave us a much richer, more accurate vocabulary to understand the language of life being spoken inside the cell.
It's just incredible. This deep dive really highlights how much more there is to gene expression than just looking at mRNA levels. Understanding translation, it seems, is absolutely critical if you want to truly understand how a cell works, how it adapts, maybe even how things go wrong in disease. It's a fantastic reminder that even these processes we learn about as basic biology are actually deeply intricate, dynamic, and full of surprises.
Absolutely. This work fundamentally changed the perspective. And maybe a final thought to leave you with. If our cells are already using these alternate routes, these hidden starting points, these flexible rules in making proteins, what else are they doing? What other layers of biological regulation, what other molecular conversations are happening right now that are still just beyond the reach of our current tools? What other languages are our cells speaking that we're only just maybe beginning to eavesdrop on?
That is a powerful thought. It really makes you appreciate the dynamic, complex world inside every one of your cells. Thanks for joining us on this deep dive into the hidden language of life. We hope you're left feeling a little more curious about the molecular wonders happening inside you right now.